Thanks, Craig. Before I begin, I'd like to give Matt Cave credit for all the work he put in to put this R13 together. If anyone's ever put an R13 together, it's a pain in the butt. I know this because my office is right next to Matt, so I heard some of the descriptors he used when writing this. But he gets a lot of credit for this, and kudos for an excellent turnout, and it's a very, very good symposium. So you've heard this alluded, and Matt asked me to kind of give a general overview of what hepatic fibrosis is in liver disease for the non-hepatologists. So I'm going to spend about half of my talk talking about that, and a lot of the references in that section will be either landmark references in the field or recent reviews for those who are interested to follow up with this. The second part of my talk is going to bring into newer concepts about ECM, dyshomeostasis, and liver disease that are, in my opinion, underappreciated. So regardless of what kind of liver disease you're studying, there is a shared natural history. There's, be it alcoholic liver disease, non-alcoholic fatty liver disease, hepatitis virus, a lot of the drug-induced liver diseases, a lot of the TASHs, they all have a very similar natural history in which they first start off with steatosis, and then they have steatohepatitis, and I can't get this mouse to work, or well, steatosis and then steatohepatitis, which is the active inflammatory phase. Shown here, this is steatosis with its macro and microvesicular fat accumulation in the liver. If the disease progresses on and the insult continues, you then get fibrosis and cirrhosis. And historically, this is cirrhosis, by the way, where you have regenerated nodules with very large ECM depositions. Historically, this has been called end-stage liver disease, and for very good reason. Generally speaking, if you developed especially advanced cirrhosis and fibrosis, you were going to probably die from complications of that disease eventually. If you characterize liver fibrosis, it's mostly, the most distinct pathological change is a robust accumulation of extracellular matrix proteins, and that it's in fact called hepatic scarring. The treatment of fibrosis and cirrhosis is palliative. We treat the decompensation associated with having a failing liver. There are lifestyle modifications and liver transplant. The point here is decompensation doesn't fix the problem. Although even if you get successful at treating decompensated liver disease, which they have, the longer you are cirrhotic, the increased risk you have of developing hepatocellular carcinoma. So this actually is a losing battle in the end. Lifestyle modification is very difficult, especially if we're dealing with someone who is morbidly obese or a chronic drinker. And liver transplantation is effective, but there are about 3 times as many people on the transplant waiting list as available livers. The point here is, really there is no FDA-approved therapy, and I can't spell therapy to save my life, to halt or reverse disease. How come after all these years of this slide, no one's caught that? We need to identify new players. So one of the things that I want to walk through here are the key mechanisms involved in hepatic fibrosis, and they can largely be lumped into 4 categories of chronic injury, impaired regeneration, increased extracellular matrix deposition, coupled with decreased extracellular matrix resolution. We'll get to each of them in a little bit of detail. So first, chronic injury, and this has been alluded to already a little bit. Now, of course, to get scarring in any tissue, to have remodeling, you have to have a source of chronic injury, and then the scar tissue comes in and replaces the damaged area. But this is not simply a passive interaction between you lose cells and then the matrix comes in. There's actually crosstalk between the cell death and the injurious mechanisms that stimulate the remodeling mechanisms. More recently, for example, work has shown that cell death itself, like the DAMPs and that Bob Roth alluded to, as well as apoptotic bodies, are direct inducers of stellate cell activation, which are the predominant myofibroblast-like cells that produce the collagen matrices in the liver. These directly will activate the cells. Also, recent work has suggested that altered autophagic responses in the liver are critical for not only whether or not the hepatocytes and other cells survive or die, but also whether or not stellate cells become activated in response to stimuli. And a lot of work recently has been done on this. And Bob Roth and others have spent a lot of time talking about 2 key concepts in this process of chronic injury, which is priming of the inflammatory cells to respond more robustly to a stimulus, and also sensitizing of the target cells, such as hepatocytes, to respond to that cytotoxic stimulus to die more robustly. Bob gave an elegant example of how sensitized cells will die to the stimulus of TNF-alpha. Also critical in altered inflammation is that there is impaired surveillance of the fibrotic response. Normally, the fibrotic response is a normal wound healing response in the liver that is surveilled by the immune response and kept to be self-limited. One of the key things that happens with chronic liver disease that transitions to fibrosis is the surveillance fails, and the fibrosis can continue. I'm not going to spend a lot of time on this slide because a lot of it has already been covered in detail and also will be covered in detail in future. Just emphasizing the point that there is a key overlap of interactions between the gut and the different cell types in the liver, including innate immune cells and also some adaptive immune responses that aren't in this slide. And these cells, the inflammatory cells, generally speaking, are primed to produce more pro-inflammatory stimuli that directly damage the hepatocytes. The hepatocytes, via different mechanisms, are sensitized to the cytotoxic stimuli and die more robustly. Both of these effects will release mediators that directly will activate the stellate cells and lead to a fibrotic response. I wanted to point out this review because it's an elegant review for immunologists who want to study liver disease. This is talking about the crosstalk of the immune response with the fibrotic response. And many immune cells directly release mediators that will activate the stellate cells or modulate their production. Also, and I already alluded to this, and natural killer cells, which have a protective role in the inflammatory phase of the disease, actually, a damaging role in the inflammatory phase of the disease, have a protective role in the fibrotic phase because they will directly induce apoptosis and senescence of stellate cells. And both in non-alcoholic fatty liver disease, hepatitis virus infection, and alcoholic liver disease, NK cell responses are attenuated. And so they do not perform this surveillance function as well, and so the self-limitation of the fibrotic response is removed. So the next thing I want to talk about is impaired regeneration, and this is a critical concept for hepatologists. Now, anyone who talks about regeneration is required to show one of these pictures, and this, of course, is Prometheus, the titan that gave fire to the humans. The reason being is, as punishment for giving fire to the humans, the gods strapped Prometheus to a rock, and a vulture or eagle, depending on whose version you're reading, came down and ate his liver every day, and his liver would then grow back, and the vulture would come back and do it the next day. What that alludes to is the fact that even the ancient Greeks understood that the liver has a unique regenerative capacity. And etiologically, that makes a good bit of sense. Of course, as will be discussed in the next section, the liver plays a significant biochemical role as a filter for xenobiotics. It has a very high capacity for xenobiotic metabolism, and it also has very high defense mechanisms. I'm not going to calculate the Nernst equation, Dean, but the glutathione concentrations in the liver are in the high millimolar range, or low millimolar range, but very high. On top of that, the liver can be viewed as a physical barrier, in such that no matter what it does as far as biochemical defenses against toxic injury, some toxins will damage the cell, regardless of where that cell gets hit. And its physical location means that it often will take a hit for the team. So it will actually purposely allow its hepatocytes to be damaged and then die, so other more critical areas of the body do not get hit. And in doing so, it would then make sense, if you have an organ that's doing that, that it needs to be able to have a capacity to regenerate on a relatively robust basis. This requires very complex coordination. Of course, it requires division of all cell types, and even terminally differentiated hepatocytes will then divide. It requires a release of mitogens. A whole slew of mitogens are involved. There are, of course, also ultrastructural and ECM changes that are required not only for the regeneration, but for the deposition of the scaffold for the regeneration to occur and expand. And coupled with that, of course, is neoangiogenesis, so the blood flow and oxygen and nutrient supplies can get to the new areas of the organ. Failure in any part of this response can, in principle, impair the regenerative response. And what has been shown time and again is that a lot of the chronicity of liver disease is associated with the failure of one of these regenerative components. And that's something that's poorly understood or not understood enough that I think we need to start paying attention to. One of the salient findings that really proved this hypothesis well was this study published in Science about 14 years ago. In this study, what they did is they used telomerase knockouts. So these animals had an inability to prevent the telomere shortening. And so their hepatocytes became senescent after very few cell divisions. And in fact, if they did a partial hepatectomy on these animals, the livers did not regenerate as robustly. When they gave an experimental fibrotic agent, carbon tetrachloride, which is commonly used to induce hepatic fibrosis, what they found is that they developed much more severe fibrotic liver injury. This has implications because what we have seen thus far is that steatosis, be it caused by alcohol, be it caused by non-alcoholic fatty liver disease, several drugs also do this, they all impair the hepatic regenerative response. So part of the chronicity of the accumulation of damage that leads to fibrosis under these conditions may well be driven by this impaired regenerative response. So in summary, what we know thus far is that there's clearly an imbalance between liver repair and liver injury, favoring the accumulation of injury. This injury also directly will activate the stellate cells and promote that fibrotic response. Now also there are key concepts of increased ECM deposition and decreased ECM resolution. I'm not going to spend a whole lot of time on this. I've already talked about a little bit with the stellate cell activation. Other fibroblast-like cells will produce extracellular matrix proteins such as especially the collagens, and this will lead to more production. Concomitantly, inhibitors of matrix resolution such as TIMP, which inhibits these matrix metalloproteases that degrade the collagen, are also upregulated and then again exacerbate the fibrotic accumulation. So again, we have an imbalance which favors the deposition and accumulation of extracellular matrix proteins. Now, I kind of hinted at this earlier, that this historically has been called end-stage liver disease. The reason that I said historically, it is now understood that it's not necessarily the case. And this is something that's quite promising to the field, that if we have a better understanding of the mechanism by which fibrosis occurs and it is perpetuated, we have an opportunity possibly to treat it. The reason that they know this now is we now have antivirals for hepatitis virus infections that actually work sufficiently to remove the viral load. And in some of those patients, their fibrosis will revert. And a big proponent of this is a guy named John Arteel from the UK. And I just want to point out one of his reviews in which he describes that in detail. But again, this is only to a certain extent, and this is only so far if the underlying cause is removed. And again, it emphasizes the need to understand what these causes are that we can develop new therapies to target this. Okay, so that's the first half of my talk. Now, one of the things that I wanted to point out is everything I talked about in the context of extracellular matrix dyshomeostasis was in the context of hepatic fibrosis. And this is a criticism of our own field. When you talk about extracellular matrix deposition in liver, most people think by default that you're talking about fibrosis. And in fact, you can make a pretty good argument that liver research is collagenocentric and fibrosocentric. And I have this trouble a lot in papers and grants. I have to put a sentence in them saying, I'm not studying fibrosis. I'm studying ECM alterations, but it's not fibrosis. And even though I say that, some reviewers have a really hard time with that concept. So here, when you look at fibrotic changes, and the matrix changes, it's a lot more than collagen going on. In fact, there is a huge remodeling of the liver in general, and all the different matrix proteins come into play. So this is not just collagen. The reason people think about collagen is because it's the easiest to see. Also, this ignores the fact that the matrix has a number of different roles, not just in scar formation. I mean, of course, it acts as an adhesive substrate. It provides the ultrastructure on which the organ is built. It also is required for contact inhibition, so differentiated cells will not divide unless they're attached to a matrix. It also will sequester and store growth factors and plays a critical role in signal transduction communication between the extracellular space and the intracellular space. And all this has nothing to do with forming a fibrotic scar. So I wanted to make an argument that perhaps extracellular matrix changes precede fibrosis, that these changes are more than simply collagen. What are these changes, and do these changes, can these changes contribute to liver pathology? And I got into this, it's kind of nice to have Jim Lyondike and Bob Roth in the room, because the way I got into the extracellular matrix in general was based on a conversation I was having with Jim Lyondike when he was visiting my lab in collaboration with Bob Roth, and it was over a beer. And we were talking about fibrin accumulation in the liver. And it was since then, I've been doing a lot of research in fibrin metabolism and Pi1 and the mediation of this extracellular matrix, so that was probably one of the best beers I've ever had. In short, fibrin accumulation is downstream from activation of the coagulation cascade. So coagulation cascade gets activated, activates thrombin, fibrinogen is cleaved to fibrin, and this is, of course, what's the main formation of a clot. And this accumulates, and it transiently accumulates usually in the tissue, and then it's eventually cleared. And this is fibrin accumulation in the liver after an injection of lipopolysaccharide. Now, we have looked at the metabolism of fibrin matrices and the mechanism by which this occurs in several inflammatory liver models. And the data I'm showing here, for obvious reasons, is the enhancement of non-alcoholic fatty liver disease caused by arsenic. I'm not going to spend a lot of time on this. There will be several good talks later that go into detail. But what I wanted to point out here is this is, again, kind of what Bob was alluding to, where you have a two-hit mechanism, where one thing that's subhepatotoxic enhances the hepatotoxicity of a second hit. In this case, the second hit is high-fat diet, and the first hit is exposure to arsenic. Under those conditions, we get a robust accumulation of fibrin extracellular matrix coupled with the inhibitor of the proteins that degrade that matrix, which is called Pi1, plasminogen activator inhibitor 1. And again, how this works is you have the coagulation system cascade activation, the fibrin matrices accumulate, this is degraded by plasmin, which is activated by urokinase plasminogen activator and tissue plasminogen activator, which is inhibited by Pi1. So induction of Pi1 will block the degradation of these clots and lead to persistence of this matrix. And this can contribute to inflammatory liver injury via several mechanisms. What I wanted to point out that this is essentially atherosclerosis. This is exactly what you find as matrix in atherosclerotic clots. And this is some work by Sanjay Srivastava at U of L in showing the accumulation of atherosclerotic plaques in arsenic-exposed animals on a high-fat diet. So we're looking at very similar mechanisms here. In fact, you could make an argument that this is hepatic atherosclerosis. This is actually something that's been well known for a while. Aaron Bachowski's group back in the early 2000s published this paper showing that very low concentrations of arsenic, this is 50 parts per billion, well below any sort of pathologic changes that you might see, were sufficient to cause significant remodeling in the liver. So what they saw is that the fenestrae that are usually present in the endothelium disappeared, and accumulation of several extracellular matrix proteins. Again, in concentrations well below the toxic threshold. Now, so it's not only fibrin, but also these, but this is something that can be done on a point-by-point basis if you have a hypothesis-based approach. What we decided to do is to try a more discovery-based approach to learn what's happening to the extracellular matrix in response to stress. And what we did here is we're doing what's called ECM proteomics or matrizomics, which I hate those omics phrases, but what the heck. And just to emphasize a point, we're doing what's called the Texas three-step extraction method. So instead of taking the tissue as a whole, grinding it up, and then looking at the extracellular matrix proteasome, what we're doing is we're doing different sequential extractions of the sample. The sodium chloride will get the loosely bound extracellular matrix. The SDS fraction will get the more complex ones. The guanylyl hydrochloride will get most of the ECM proteins, the basement membranes. And the pellet that's left over is highly cross-linked extracellular matrix proteins. And this is the distribution of ECM proteins from a control liver in these differential fractions. So you can see it separates out the proteins quite nicely into different groups. The reason that we do this is the maturity of the matrix proteins comes also into play, not just the amount or the accumulation, but also what state they're in. So for example, plasminogen, if it's present, it accumulates in the same fraction no matter what. However, if you look at annexin, in the controls, it's present in the pellet, so that means it's highly cross-linked. It disappears in response to the first two treatments. In the fourth treatment, it's back again, but it's in the first fraction, so that means it's probably newly formed or in high turnover. Now, fibrinogen is my favorite example because if you looked at total liver extract, you'd say, okay, fibrin's not changing. But if you look at these differential extracts, you can see that there's a lot of activity going on with those fibrin ECMs as far as long age and metabolism. And so this is very interesting and very useful information. And just to hammer that point home, what we have here is the effect of ethanol diet for 6 weeks, or the effect of lipopolysaccharide, 1 dose, and harvesting the livers 24 hours later. And I'm not going to go into the weeds on this one, but I wanted to just show you that the ECM responds dynamically to stress, and very dynamically. And if we compare the overlap of ethanol diet, LPS, or the combination of ethanol and LPS, again, another kind of two-hit model, we see we get a nice stratification of the different ECM proteins into different groups. So we have now developed this technique to leverage questions about what's happening to the ECM proteasome in detail. Now, one of the things that we still need to question are, do these changes in the ECM mean anything? Do these contribute to pathology? And based on some in vivo work we did a few years ago, and also work of other groups such as Bob Ross and Jim Lyndykes, we already have that proof of principle down, which is that ECM changes will contribute to inflammatory liver damage. And here's a good example where we did a two-hit model of ethanol and LPS, and we saw significant fibrin matrix accumulation. If we blocked that matrix accumulation by using either TNFR1 knockouts or antibodies against plasminogen activator inhibitor 1, we block the accumulation of this matrix and we block damage. What we are interested now in doing is, again, trying to do it more systematically in vitro so we can get more new information. And this is basically extracting the extracellular matrix from a liver, and it's actually really cool looking if you've ever not seen it. So this is a liver before decellularization. After decellularization, it looks pretty much like a liver ghost. You take that matrix, you dry it out, grind it up, and then you use it as a substratum for cell culture. And when you do so, what we found here is this is the effect of taking control extracellular matrix and looking at the production of pro-inflammatory cytokines by raw macrophages. The control matrix has no effect over just normal raw cells grown on plastic. If you take the matrix from ethanol liver and put it on plastic, you see a robust increase in the production of several pro-inflammatory cytokines, which, if you've been paying attention in the past few talks, are known to be critical in the mechanisms of inflammatory liver damage and the progression of liver disease. So we believe now we have in vitro a model in which we can start looking at these matrix changes and seeing what's in play and what the key mechanisms are. So in summary, about hepatic ECM homeostasis and liver disease, and that's why I had this little gray liver on the first. That's my version of the liver ghost. So fibrosis, of course, is well known to have dramatic remodeling and scarring of the liver. It's mediated by an imbalance between injury and repair, and synthesis and degradation. It is potentially reversible, hopefully with better understanding of these mechanisms by which this occurs. We can develop target therapies to enhance the normal ability of the liver to resolve from damage. Also, one of the things I wanted to emphasize is the ECM changes dynamically in response to stress. Within 24 hours of a stressful event, you see significant ECM changes. This is normal, and this is well understood in other organs, but because of the strong interest in hepatic fibrosis, the liver field has largely ignored it up to this point. In fact, the impact is poorly understood, and this again may be an effective target because if you can block The transition from steatohepatitis to fibrosis and cirrhosis, that would already be considered a win on many categories. If you can enhance the reversion of steatohepatitis to normal liver, that would be a significant win, and certainly a goal to better understand the mechanisms by which this occurs. And of course, nothing is done in a vacuum. I'd like to give credit to my laboratory and some of the people who did this work. I showed work by Christine Dolan, Veronica Massey, and Lauren Poole. Some other recent arrivals to the lab and some former lab members. Of course, my collaborators, Ileana Bayer, Jesse Roman. Michael Merchant is the proteomics guru that helped me with this. And of course, Craig McLean, and grant support from NIAAA and NIHS. And with that, I'm happy to take questions. Thank you.
Questions for Gavin?
Very thought-provocative, Gavin. Two questions for you. I take it that you're implying in your message that ECM remodeling is preceding the classical pathological fibrotic phenotype downstream.
Correct.
Is that a correct assumption?
Mm-hmm.
So have you looked longitudinally to look at the composition of that matrix, how it evolves over time?
So that's an elegant question. We need to do that. We have only, I mean, the data that I showed are literally a couple weeks old. So, but I mean, having this technique in hand is then something we can do that. To the first question, yeah, there are several studies showing that matrix changes will precede the inflammatory response and may be part of the sensitization. Good examples are what's called the alcoholic lung phenotype, where the alcohol exposure will cause matrix remodeling that will precipitate an inflammatory response to like pneumonia. But if you look at the liver, at the lung itself, there's no damage per se. And that's also true for liver, and of course all the work with integrin and anti-integrin inhibitors are playing into this hypothesis that the matrix changes are critical for sensitization and priming.
Now actually your answer sets a good stage for my second question, which is as you begin to sort of look at this more mechanistically,
Mm-hmm.
I presume that you're going to be looking at integrins, because obviously ultimately that's going to drive the biology of the system.
Mm-hmm.
And so it will be really interesting to see if the either partnering or the deposition of these integrins on the membranes of target cells
Mm-hmm.
are going to be dynamically responding to the evolution of matrix changes.
That's an elegant point. In fact, I didn't show the data here, just but in that two-hit model in vitro that we did, that the ethanol matrix increased alpha V beta 3 integrin expression in the raw macrophages in response to stimulus. And the experiments you are suggesting, my student Lauren is doing, I hope, next week or the week after. So we'll keep you posted.
Gavin, really nice presentation. I really like the new techniques that you use in generating this ghost delivery. My question deals with your extracellular matrix when you culture the cells with dastrin-controlled and alcohol-treated animals. The question is, are you thinking that there are newly synthesized proteins in the alcohol matrix that is giving you this differential profile in cytokines, or perhaps the 16 proteins that are being modified? Oxidized thiols, things like nitrated proteins, things of, so what's your thinking as to how this extracellular matrix from the alcohol-treated animals, what is in there that is giving you this differential profile?
Okay. The short answer is we don't know. The long answer is based on precedent in the literature and what we know thus far, for example, osteopontin, vitronectin, fibrin, fibrinogen, we suspect that, and all those ECM proteins, of course, are up in that matrix. So we're suspecting that it's going to be new proteins that are found in the matrix that are driving this. However, there are also suggestions, for example, that it's not only the 2D interest, but also the uncovering of matrix proteins that may play a role. So it could be also the proteins that are there but look different. So where they are in the extractions will be of interest for us and very important too. Good question.
Okay, I think we better stop now. We have a 15-minute break, it looks like, and then we're meet back here, Matt, if I'm not
Yeah, so
mistaken.
we have about a 15 minute.